Okay, so we're to the point where we've covered pretty much all the material that we're going to get to this semester, and the final exam is coming up. So what I'm going to do is just do a very quick review. I'm going to try to keep this as short as possible, but yet I'm going to try to cover an overview of everything that's on the final exam from the fourth block of material, and a few of the things that are cumulative from the first three exams. And I'm going to try to do it in less than 30 minutes. Just to give you some focus as you study for the exam, especially with the cumulative part, which I'll get to then toward the end. So the fourth exam material really is on, there's a little bit on Archaea. So the Archaea material you should, so part of it is Archaea. You should just know a little bit, of the groups. Their metabolism is what's very unique: ammonia oxidizers, methane producers, sulfur reducers, and so on. But they're still categorized by the 16S ribosomal RNA. And then they get names, and those names, where they are, whether they're phyla or whatever, is changing all the time. So it's not of a paramount importance that you know exactly all those details. But I went through in a little lecture a couple names of some groups and some of the key characteristics of those groups that you should be able to know just to kind of match them up to identify. Really, though, the bulk of the material has to do with both viral infection and then bacterial pathogens. So these would be pathogens. And then the response of our cells. So the human immune response. So that's what I'm going to kind of focus on this as a kind of an overall summary of this review. And then I'll get a little bit more into afterward some of the cumulative topics. Many of them will kind of feed into this anyway. So started off with, say, a virus. The virus can be naked, or the virus can be enveloped. And I gave you some examples, of these. So even this guy would have little binding proteins. Either way, the virus has a capsid that is a protein shell of some sort. Icosahedral, we said, was very common, but there are helical ones, as well. Those proteins protect the genome, whether it's DNA or RNA. It could be double-stranded or single-stranded DNA or RNA. Outside of the viral capsid, there could just be proteins that would bind specifically to receptors on the cell membrane. They could be part of the capsid like this, or they may be attached to a membrane, which is the envelope. Now, what will ultimately happen is the virus will bind to will bind to a host cell. Wow, it's really looking bad there. Like this. And it's usually fairly specific. So a virus is often even named by the host. So you have animal viruses, and plant viruses, and then bacteriophage that are just specific for bacteria. Bacteriophage do not typically infect a eukaryotic cell. They're specific for those prokaryotic cells. Plant viruses don't infect animals. So each of them will have very specific binding, and even within animals, there are viruses that affect certain groups of animals and not other groups of animals. So it's more rare than they start to jump groups of organisms, and then even when they're in a group, say mammalian virus, often they might be specific for a species and they just don't affect other species. So those that actually jump between different species then become of more concern, especially if they're mammalian and can affect humans. We have an interest in them, obviously. And that's because they're specific binding. So that's one of the first things. They bind, and then they get into the cell one way or the other. They either get the actual viral particle, the capsid comes into the cell, either directly, or it comes in through an endocytosis process, or through a merging of the envelope with the cell membrane, or alternatively, it's just the genome, that gets into the cell. depending on what it is, this is where that Baltimore System part comes in. What I'd like you to do then from that is looking at the Baltimore System, depending on what is the starting point. So what type of genome is this? Okay, is it double-stranded DNA? So it's going to be our most generic and straightforward. But that's not necessarily the most common. Okay, so there's single-stranded DNA. There is single-stranded RNA plus, single-stranded RNA minus, and then we have the retroviral, which is also an RNA, and then there's different type of double-stranded DNA virus that goes through a sort of a different life cycle process. So the life cycle of a virus, regardless of what it is, several things have to happen. So we need to replicate the genome, and then we have to express the viral proteins, which will remake a new capsid. If there's proteins associated with an envelope formation, there'll be envelope proteins that will be produced as well. But the structural proteins, so they replicate the genome, and then there's structural viral proteins. And then the whole thing gets assembled, and then it'll eventually leave the cell and spread. And then there's different ways in which that will happen. Okay, so it could bud and take a piece of the membrane with it, or it could cause the cell to lyse and burst, and then the viral particle, is coming out through that bursting process and would then go on to infect other cells and continue the cycle. So we have lytic and lysogenic cycles. In the lytic cycle, it's going to just replicate the genome, it's going to replicate the viral proteins, it's going to assemble them, and typically the cell will burst and die. In the lysogenic cycle, so this could happen in two ways, lytic or lysogenic. In the lysogenic cycle, what will happen is the genome may then get into the host cell's nucleus, which does have to happen in some of these cases anyway, in eukaryotic cells, and it can actually join into the host cell's DNA. So if the host is, here's our DNA, and then the viral genome, gets kind of spliced into it. So that's actually viral DNA stuck there. Now, obviously for this, that would work for the double-stranded DNA virus, but there are other types of virus that do this that don't start off with double-stranded DNA. So that means the RNA then has to be reverse transcribed into DNA. So HIV does this. And then the DNA would just be a single strand. There'd have to be a double strand, and then they can actually join in. So there's all kinds of possibilities. Pretty much all the possibilities are possible. It's just that the logical step material that was on our third exam, from DNA to RNA, RNA to translation to a protein, is going to still follow all the same rules. There are not new or different rules or a breaking of any of the rules. It's just that you're often going to have different starting points than only double-stranded DNA. The starting point could be single-stranded. It could be RNA as a starting point, but then you may have to go to DNA to come back to RNA. So you should be able to kind of walk through those processes as an outline. What would be, the first step in replicating the genome, and then what would be involved in translation or getting the structural protein. So, for example, if you have the single-stranded RNA plus, that typically is messenger RNA, and in many cases it actually even has a 5' cap and everything with it, so it's ready right when it gets into the cytoplasm to have ribosomes bind to it and then read it and actually start expressing proteins. Some of those proteins will be a replicase protein, which will then start copying the genome. First you have to make a template to work off of, so that the antisense strand, and then that will just be the template for making more and more copies of the RNA plus, the one that you can actually read. Some of those will act as messenger RNA, so more translation will occur, and some of them will just be the genome that will eventually get packaged up. Ultimately, more of the structural proteins, the later bond proteins will get made, and then there'll be the assembly and everything else. So go through what's the lytic cycle, be able to outline it. What's the lysogenic cycle? be able to outline it. For different types of virus, also be able to say how they would get their genome replicated or how their structural proteins would be translated from their gene, so the gene expression process. And then just the very generic sort of the assembly of all this, the process for that, and then the leaving of the cell, which could be in a variety of different ways, but just kind of that concept of what would be the difference between lytic and lysogenic. So review all the parts of the viral life cycle and the differences between different types of viral genomes. Now, eventually we'll get to, say, human immune response to this as well. Something to keep in mind. Well, actually, I'll put it in right now. We'll kind of pepper that in little by little. So, some cells in your body will have been exposed then to a virus at some point in time. And what will happen is those cells, say for example, like a B cell, may have an ability to bind to this virus. Right? And it recognizes it now as something foreign that's not part of one of your cells. Right? And so what it will do is potentially, a number of different things. The B cell will then be triggered by this organism to ultimately make more copies of this particular binding protein. Okay, so the virus who's different or unique, those proteins on it would be called our antigens. Not our antigens, it's the viral antigens. Antigens are things that are foreign to our body. The proteins that match up with the antigens are called antibodies. And what a B cell can do is, the thing is, you keep this in mind, there are almost an infinite number of different types of these proteins on B cells. There's all these ones, and some of them have never encountered the particular organism or antigen that they would match up with. But when they do, it triggers a response. It wakes this B cell up, and one of the things it can do, is replicate more copies of it. This binding will then release chemicals, okay? So these cytokines, which will then trigger more of the immune response. So they'll bring in other cells to phagocytize the foreign particle and destroy it. There are some chemicals that can just straight away destroy the molecule. There's all different types of cytokines, and they cause inflammation responses and all these sorts of things. So they'll bring more immune cells in, they'll bring different immune cells in, and they'll eventually try to destroy the foreign particle or organism. But in addition to that, this B cell can form a plasma cell. And what the plasma cell will do is it will make more copies of this antibody that it will ultimately release. So these would then be free copies of the antibody. They're not actually attached to the cell. Which means that then the next time that you get exposed to this virus, these antibodies attach to its surface. So now the virus cannot attach to one of your other cells, to that receptor, because those little binding proteins are already bound up. Okay, this is a process called opsonization. I'm pretty sure I cover this in another lecture. And it's an important part of how the immune system responds to bacteria. It does it this way too, and virus as well. So this way it does two things. This viral particle now cannot infect the cell. It's also tagged, so phagocytes will come in and then try to eat and destroy that particular viral particle, so it won't be able to infect actually any of the cells. Right. So that's kind of how the immune response comes in after the fact. The next thing we'll look at quickly here then is a bacterial infection. Okay, so a bacterial pathogen. And this is important because, and I'm just going to make it look a little bit different. Here, kind of make an extra layer for cell wall, like this. And we'll put a little circular DNA in it for the genome of the bacteria. So this is a bacterial pathogen. So after you're done going through everything with the virus material, then you need to start to look at the bacteria that are pathogens and the process. So there's going to be an adhesion process. Same thing. All right. There are going to be some ways that a bacteria will bind to a host cell. So the bacteria itself will have glycoproteins on its surface that could bind to certain types of cells, to specific receptors on the surface of them. Sometimes the binding can be more generic, and other times it's highly specific. It really just depends on the organism as a pathogen and its mechanism. But there's some way in which there is a binding specifically, so an adhesion between the bacterial cell and a host cell that's part of a particular tissue. What will happen is then the bacteria can do a number of things. It'll also start to release some enzymes in the environment. And if there's a tissue, say for example, where this is not just a single cell, but it's part of a group of many cells, here, that the bacteria can release chemicals that can start to break the bonds like between multiple cells, or it can cause the lysis, of a cell that will start to then release the nutrients of that cell. So the bacteria has its own machinery to replicate itself. It's one of the big differences here between the bacteria and the virus. The virus, one of the main reasons why it is infecting the cell, is because it does not have the ability to express its own genes. It has no ribosomes, and it doesn't have all of the enzymes it needs to replicate its genome. Okay, so it doesn't have the DNA polymerase. It doesn't have these things that it needs to make more copies of itself. It needs a host cell, a live cell, in order to do that. It may ultimately kill that cell, but it needs the cell to be alive for it to be able to be replicated. The bacteria don't necessarily. There are some bacteria that are intracellular parasites, so they may go into a cell when they cause the infection. But many times the bacteria really just wants the nutrients from the cell so it can replicate itself. Okay, so the bacteria, through our binary fission, right, can do its own cellular division and replication, its own DNA replication, its own gene expression, all things we went through, the bacteria does itself, but it needs nutrients in order to do that, all right, and raw materials. And that's what it's going to be potentially getting from a cell. So these enzymes become important because we're releasing contents of cells and tissues that are then feed the bacteria so that they can divide. All right, that's kind of the bacterial pathogenesis. So they bind to surfaces of tissues, specifically then to cells, of those tissues. They release their enzymes to damage or destroy those cells and tissues. And so there's examples of ones that can kind of break the tissues and cells apart. The hyaluronidase that breaks down hyaluronic acid in connective tissue so that the bacteria can actually work its way in, sometimes to the tissue, like this, where then it can start to multiply. So then we get more copies of the bacteria as they're dividing, and then they divide, and then they divide, and they divide, and they keep dividing, and we get a lot more bacterial cells causing an infection. Right? So you get the adhesion of them, the release of the particular enzymes. Now at the same time this is happening, what will also occur is that the host immune system is going to be responding to them. So you also have now these cells that are macrophages. They're kind of generic. They're cells in your body, part of your white blood cells. They're going to look for things that are foreign, and if they find them, they'll eat them. They'll just try to phagocytize them. Okay, so they would take that bacterial cell and try to eat it, so they can ultimately bind it with one of their lysosomes. So a lysosome would be like this. These things would bind together. The acid and low pH environment of the lysosome would then potentially destroy the bacterial cell, the pathogen, or any other just invading organism that's not supposed to be there, and get rid of it. to link with other parts of the immune system, some of these phagocytic cells are called dendritic cells. And a dendritic cell, after this process occurs, what it can do is it can actually take some of these pieces of the bacteria, the ones that had these antigens that were these foreign things that the bacteria is going to use to bind to cells. But what it'll do is it'll actually pop them up on its surface. So they become APCs, so an antigen presenting cell. And they can then interact with other cells in the immune system to trigger future immune responses, future sort of preparatory immune responses, so that if we haven't seen this bacteria before and we get exposed to it, we then kind of build up that sort of immunity to it by creating more antibodies and certain cells that can then recognize it quicker in the future, things that are more specific for it, as opposed to being sort of more generic. In addition to that, these cytokines, some of these are antimicrobial agents. So some of these chemicals, as they get released, will actually try to kill the bacterial cell. So there's a whole bunch of processes that are going on in your immune system. So where there are cells that are going to release chemicals that are going to try to destroy the bacteria, there's the whole immune response of inducing fever, which will increase temperature and then pull iron out of the environment in the tissues. And the bacteria need the iron in order to do binary fission and divide. So essentially, fever will starve the bacteria. The heat isn't what destroys them. Some people say, oh, the heat would denature the protein. That's not it. It's the iron depletion that will actually cause that to happen. So you need to go through the steps of bacterial pathogenesis, and the real important thing that I want to stress more than anything else is, as you look at, I have a few lectures on that where I go into details. This is just a kind of overview. Make sure you recognize it as being something different than the viral infection. They're two different things, okay? And many times I find people know a lot of detail, but they're telling me all the detail about a viral life cycle. They're not talking about the bacterial infection. Or I ask about a viral life cycle, and people are telling me more specific things that have to do just with a bacteria. It's not a virus. So make sure that that's kind of the main things as you're focusing on your studying. Keep these separate. Viral infection process, what is a virus, how it works, and then bacterial infection and how that works. And then at the end of that is the immune response. Immunology is a very typically a high level. Usually it's a 400 level course for undergraduates or a graduate level course. It's typically not taught at a lower level because it is very complicated with the biochemistry of it, the different types of cell biology and genetics involved in triggering the different processes. So you really just need to know kind of the overview and some of the things I've talked about in a few of the different lectures that I have on the initial responses, sort of the innate responses that we just all have to things that are just generic. And then there are some overview of the specific responses which deal with the B and the T cells. Okay, so make sure you know what's a B cell, what's a T cell, what do they do in general in terms of the response. There are all different kinds of them, and it gets into a lot of detail, so you should know the definitions more of terms when it comes to that. Okay, so when it goes to human immune response, you have barriers like your skin. The skin is salty. The skin is a more acidic pH it can have. It's things that are just part of you that will just stop certain bacteria from being able to infect. But if you have a cut or a wound and the bacteria can then get in and start attaching to the cells and start releasing its enzymes, it's going to start to begin the infection process, and then you're going to have more of a cellular response. So some of the cells will have to be producing chemicals, the cytokines, releasing them to try to stop that organism from growing or to kill it. Okay, you'll have cells trying to eat or destroy the phagocytes. Some of those cells will then consume them and then take pieces of them to trigger more of an active immune response that will then create specific cells for that specific pathogen, which will come later. And then, like I said, it gets very, 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 very deep in terms of the concepts and all the details. But if you have an overview of those processes, the main sorts of things I talk about in the videos, even those are just very much of an overview. There isn't even a lot of detail there. Before we run out of time, so that's kind of the main material that's on this exam that's new. And that'll be the bulk of the exam. So it's 150 points. 100 of the points will be mostly this kind of material, the multiple choice questions, and some of the essay questions that you'll have to do and turn in. Cumulative. All right, our first exam had to do more with bacterial structure, so things like the cell wall, peptidoglycan structure, and the differences between gram-positive and gram-negative. That's really going to be the key there. So make sure you can define peptidoglycan, what makes up a peptidoglycan, and what is the difference between the overall cell wall of a gram-positive, gram-negative bacteria and just the overall structure of a bacterial cell. So things like periplasm, things that are unique for a bacterial cell, fimbriae or the flagella, just in general, what are the structures that bacteria have? That was a lot of what was on the first exam. Second exam. So you're going to have like one essay question related to this sort of thing and a few, maybe 3 or 4 multiple choice questions related to that first exam to give you a feel for how much material. Second exam we went into was more metabolism and cell growth. So here you need to kind of review things like you have glycolysis, pyruvate oxidation, citric acid cycle, electron transport chain, and so on. Generally those processes, so metabolism in general, just do a quick overview. You're not going to be asked really detailed specific questions about all of those things or like finely detailed things about this particular step in glycolysis. You're not going to have those kind of questions. It's more of an overview, of things that were particularly important. And I would say one theme throughout all of that material has to do both with producing ATP and the importance of NADH in producing the ATP. So our oxidation events. So essentially, nutrient molecules are oxidized, which means the electrons are taken away from them. The electrons are carried by the NADH, which is the electron carrier, and those electrons are used to power the active transport pumps that are in part of the cell membrane of the bacteria, pumping. So this is now a bacterial cell. Bacteria cytoplasm. Bacterial cell wall. We have a space, remember here, a periplasm. And then embedded. And this membrane is where the electron transport chain is, okay, in the actual bacteria membrane. Right, and so the NADH delivers electrons to these pumps, which fill the periplasm then up with hydrogen ions or protons. The electrons go to oxygen. Right, so oxygen drives the electron transport chain. It's an aerobic process. And then all these hydrogen ions will ultimately return into the cytoplasm of the bacterial cell. But they'll return into the cytoplasm through diffusion. And it's that process of diffusion which gives off the energy that the cell could use to make ATP. The NADH is really the key. And so another aspect of this that we had on our exam was fermentation. Which is essentially where we don't have oxygen. So this process can't happen. All that can happen is glycolysis. So this is why it's important to know a little more about glycolysis, because it's occurring with or without oxygen. It's always occurring. The Archaea do glycolysis. All the bacteria do glycolysis. Our cells do glycolysis. Why should you know about it? Well, pretty much everything that's alive needs it and does it all the time, for the most part. So fermentation then allows the cells who don't have oxygen to continue glycolysis, because what eventually happens is to make NADH, you need NAD+. The NAD+ is what picks up the electrons through the oxidation events. It gives the electrons to these guys, and they become the NADH, who deliver the electrons to the electron transport chain. But now, if there is no electron transport chain, then these guys can't deliver those electrons anywhere, and so they just sit around and do nothing. And that means ultimately these get used up, and we have none left, so we can't even make any more of these, which means the processes in glycolysis all stop, and the cell can't make any ATP at all. All right, so you need to always have a supply of the NAD pluses, and that's what fermentation does. So the purpose of fermentation is to kind of recycle the NADH back into NAD plus when there's no oxygen. Okay, so if there's no oxygen, fermentation will do that and allow glycolysis to run. That was a really important part of tying together some of the metabolic information from aerobic and anaerobic that was on that particular exam. So make sure you know some of the things about fermentation that we covered, specifically things about NADH and NAD plus NADH recycling to that process, and the NADH that are made in some of the processes of metabolism, like glycolysis, pyruvate oxidation, citric acid cycle. So not all the enzymes, not all the steps, but kind of the oxidation steps, those sorts of things which carry a large level of importance. Okay. So that will be your focus for the second exam material. That stuff right there. And then for the third exam, which wasn't all that long ago really, we have DNA structure, DNA replication, and then gene expression. And that, again, there's a huge overlap then of what we went over in the virology material, because for a virus, what you need to know very specifically is how is their genome replicated, how are their genes expressed through gene expression. You have to go through those processes. So you absolutely must need to know this, and there may not be really any specific questions that are like third exam alone, because there's so much overlap between them and this material. You're going to be asked those questions, and they're kind of both viral replication related and just generic, like what is the process, how does the process work, that sort of thing. The main thing from that third exam that was different or unique from this was the horizontal gene transfer. So make sure you then go back as something that's different. It's not part of this material really. Well, actually it is a little bit with our lysogenic cycle, right? We have transduction. Right, our specialized transduction with the lysogenic cycle, right, where when this happens and the genome goes into the bacteria, that can be cut back out eventually for its own replication. And when it's cut out, coming along with it is a piece of the genes of the host organism, which can then allow more like recombination. So in bacteria, so again, we're talking about for horizontal gene transfer, this is a process not occurring in eukaryotic cells. This is a bacterial process to increase their overall genetic diversity. Okay, so they're getting genes from other bacteria from a virus. That's the idea of the transduction process. So, but know things about conjugation as well, because we said that was an important one, and the transformation, just with a definition, like what's transformation? Getting DNA from the environment. So make sure you know about horizontal gene transfer, because that was an important process for bacteria. Go over that. F plus cells, F minus cells, make sure you know what they are. What's the pilus? What crosses over from the pilus? So the single-stranded DNA. So review that material. So I would say focus on horizontal gene transfer a little bit, and then this DNA, RNA, gene expression stuff, kind of there's so much overlap that you need to be doing that as you go through the Baltimore system of viral genomes to kind of find how do they replicate it, how they do it. You'll be covering any and everything you need to know as you're going through that material there. So mostly just a little bit of Archaea, just maybe 2 or 3 questions on Archaea, not a whole lot, just the names of the groups, a little bit in are they ammonia oxidizer, are they a methane producer, that type of thing. Virus, there'll be maybe a few questions of an example of like who is on your study guide. There was some matching, like Ebola is this type of virus and so forth. And I did do a little coronavirus lectures in addition to that. So it's an enveloped, single-stranded RNA plus virus, and so on. So I might just give you a little matching type thing with just 2 or 3 of those. But in general, it's a viral life cycle, and then comparing those. For bacteria as a pathogen, I want you to know those processes of adhesion and penetration into the tissues, and then spreading factors. And again, one of your readings lists some really nice information with big tables, which is tons of enzymes and all the things they all do. I gave you in the little lectures a few samples of them. That's all we would really need to know is the couple examples and samples of that I told you about. The rest of it is there, though, as a resource for you. And then the immune response, again, is a massive, potentially complicated thing, but we're just looking at the basic innate responses, barriers and stuff at first, and the first cells that come on the scene, and then what in general is the role of the B and the T cells afterward. Cumulative, focus on kind of cell wall structure for the first exam material. Second exam material, fermentation, NAD, oxidation events. That would be the main thing to focus on there. And then horizontal gene transfer will be the main unique thing to focus on then from the third exam material. Obviously, that's a given, the DNA RNA stuff. I think that covers all of it. I have the exam. The exam is done. It's actually already posted in Blackboard. It'll just become active at Monday at 1 a.m. So Monday the 11th at 1 a.m. it's active, which means you can start doing it. Not that anybody's going to do it then, but that means first thing on Monday, whenever you're awake, you'll be able to print off the written part. So you can start to work on that, and then you'll have the online part, which you'll have 1 hour to do, 60 minutes, whenever you do it. So you could do it on Monday or Tuesday or Wednesday or Thursday or Friday. It's going to run all the way to Friday, essentially 11 p.m. That's when it'll end. So Friday 11 p.m. it'll disappear. So that's the end of finals week, essentially. So you need to do it. You need 1 hour, sometime within the week, to sit down with no distractions, and be fully prepared to answer the questions. And if you need resources or want them handy, have them there. You're not going to have time to look everything up. Make sure you've studied and watched videos and read beforehand. And then you should be able to finish it in like 20-25 minutes, really. And if you do, then you potentially have a little bit of extra time to check, make sure you answered the questions correctly. There's also the written part, which has some sketches and some things to explain. Essentially, you just have to, like you've been with other assignments, turn that back into me so email it to me, before Friday at 11 p.m. So again, you could do it all on Monday and just be done with it, the written and online part, and that's it. Then we're basically done. Turn in your other assignments. I'll keep accepting any other things with labs or any other material up until the end then of day on Friday. So they don't give us actually much time. It's actually Monday. I pretty much have to have, by the following Monday, I have to have your grades all done and turned in. So that the weekend is the only time I can actually grade any of the written stuff that you turn in. So if you turn in literature assignments, the last one, remember I posted that's going to be extra credit now. So if you run out of time and are panicked and say, I forgot and I can't do it and I didn't realize it was due, don't actually worry about it. If you have done it or are working on it or have the time to do it, then you can do it and you're going to get extra credit for that. And if you haven't done some of the labs, just get them turned in. Most of you have done pretty much all the lab material and done a good job with it. So that will be it. Okay. And if you have questions, send me emails and I will keep looking for them, especially through the week. If you have technical problems, just don't panic. I'll get it. I'll fix stuff as soon as I can. This morning, for example, I cannot get into Blackboard. So I wanted to post something, but I can't do it. So I understand you may not be able to get in at the time you want and it may mess things up for you. I'll be flexible and try to fix it whatever I can. All right. Good luck.